Hi, my dear students. Myself, Aneesh. I'm from Excel Educational Center, and here I am with another episode of Rapid Focus series. Okay, and my dear students, I think all of you are doing well. And here you can see, and today I am here to discuss some important questions related with the chapter Life Processes. And you know that in grade 10, as you are very familiar, there are 5 chapters in biology. And the science whole together, 80 marks, and out of 80 marks you can see, and 30 marks from biology. And biology itself, there are 5 chapters, and the most important chapter, the heavy weighted chapter, is nothing but the life process, my dear students. You know that you studied this chapter many months back, so that's why this is a little bit confusing, as well as it an elaborated chapter. The reason is both plant physiology and human physiology are combined together in the topic life processes, my dear students, because as the name indicates, the processes which are very essential to maintain life are together called what? Life processes. And you know that we are all living organisms. When we are considering the planet Earth, and it's also called the biosphere. And why it's called biosphere? Because life is there only in the planet Earth, you know that. That's why the planet Earth is called a biosphere. And that biosphere, you know that there are different types of living organisms are there. And that living organisms are maintaining their life, you know that. And for maintaining their life, you can see, and there are some life processes are there, my dear students. And here you can see there are 4 different life processes are there. One is nothing but nutrition. Nutrition, you know that the word is everything related with what? The food intake and the complete utilization of each and every component of the food. For example, the living organisms like plants, and they are performing photosynthesis, and they are obtaining, or because they are preparing their food, they are coming under autotrophs. And the other organisms are called what? Heterotrophic organism, just like human beings. And we are all heterotrophic, and we cannot produce our food. But what we can do, we can consume it. And we are heterotrophic organism. We are obtaining nutrients from other living organism. And I am not giving a detailed idea about the chapter. It is not the right time, because you are all waiting for your board examination, and it is on February 20th, my dear students. We are going to appear for our board examination in 2025. And here you can see what are the things you have to learn from the chapter life process. And here we can see some questions related with that. Okay, so we can start our questions. And here I include MCQ type of questions, assertion and reason type of question, as well as the descriptive type questions. These are the 3 different types of questions you can expect from the chapter life process. We can do one by one. The chapter again, grade 10th life process. Listen, my dear students, the first question. And here you can see there is a question is there on the top. List out any 4 adaptations. The word adaptation is very familiar to you. And each and every organism is having their own adaptations are there. And here you can see list out any 4 adaptations of leaves. Hey, list out any 4 adaptations of leaves for photosynthesis. Actually, we studied different other adaptations we studied. But this is an important topic, right? And list out any 4 adaptations. And when you are considering a leaf into account, leaf is known as the kitchen of the plant, right? Leaf is known as the kitchen of the plant. You already studied the structure of leaf. You studied already. And here you can see, when you're looking at leaf, you can see, here you can see the leaf is always attached on a stem, you can see. So this is the stem, and the stem is having a node region, NOD. It's not important actually, a leaf, and a green in color. And you can see why it is green in color. You know that the green in color is due to a green colored pigment is there. And what is that green colored pigment? It is called chlorophyll. And chlorophyll is green in color. That chlorophyll is the pigment which is present in the leaf, and that is very essential for absorbing the celestial source of energy to the planet Earth. That is nothing but solar energy. And the morning time or the during daytime, the sun is emitting the sunlight, and from the sunlight, only the visible part of the sunlight. And you can see the electromagnetic spectrum you studied, my dear student. In electromagnetic spectrum of sunlight, only the visible part, and nearly from 400 nanometer to 700 nanometer, that is a visible spectrum of sunlight. And that only that visible spectrum of sunlight is used by the plants, and that is called the photosynthetic active radiation, PAR. The photosynthetic active radiation, the PAR, is absorbed by the green colored pigment called chlorophyll. The chlorophyll can absorb the sunlight, and the chlorophyll can convert or can absorb, as well as you can see a mechanism is there already designed inside the leaf, which is responsible for converting the solar energy into the chemical energy. A conversion of, you already studied, you can see energy can be neither be created nor be destroyed. It can be transferred from one form of energy to another form of energy. You studied in thermodynamics. And here you can see the sunlight, the solar energy is converted into chemical energy by a process called photosynthesis. And photo, as you all know, that's sunlight. And here you can see the leaves are the sites of what you can say, the photosynthesis. They are known as the kitchen of the plant. And we are taking a leaf into account. Inside the leaf, you can see different components are there, especially upper epidermis, lower epidermis. In between upper and lower, you can see a particular type of chloroplast. Chloroplast containing cells are there. The chloroplast containing cells are called what? The mesophyll cells. And the mesophyll cells which are attached on the top are called what? Palisade mesophyll. And palisade mesophills are located just under the upper epidermis. They are tightly packed with enough number of you can see the chloroplast, you can see. Anyway, and we can directly move to the question. Here you can see what are the adaptations, and we can discuss, my dear students. And the first adaptation, you know that when you are looking at plant, you can see a leaf is having a broad surface area. A leaf is having a broad surface area, and that is the first point, my dear student. The leaf is having a broad surface area, and you know, broad surface area means what? Otherwise, what will happen? If you just imagine, if the leaf is a thread-like structure, narrow thread-like structure means what? It can absorb minimum amount of sunlight. So that's why the leaves are open. And you know, we studied no touch me not plant drooping. It's not drooped actually. The leaves are open, my dear student. So when the leaf is open, you know what is that broad leaf outer surface is called what? You study that name, my dear students. I think you're remembering, some of you are remembering that word, and that is called what? The leaf blade. Otherwise it is called what? Lamina. So the leaf is having a broad surface area, and you know, broad surface, broader the surface area and more the absorption of sunlight. You already studied alveoli inside our lungs, and broader surface area, more exchange of gases. Hey, you studied right. And like that, you can see small intestine is fully enriched with a small finger-like projections called villi. And villi means what? You can see the broad surface area. Broader the surface area, more the function. Like that, you can see the leaves are broader. Broader in the sense, what is that? The lamina is broader. So that's why they can absorb more amount of sunlight. That is the first point you have to mention in the board examination. Whenever there is a question, you can write point-wise, my dear student. Broad surface area for the absorption of sunlight. Okay. And the lamina, you should mention the name. Okay. And the second one, you know that when you are looking a plant into account, you know the upper surface of the leaf and lower surface of the leaf, and both are not same in color, right? Color in the sense, color is green, but the intensity of the green color is different, right? Why I already started that point actually. You know, when you're taking a leaf, you can see the upper surface of the leaf is having palisade mesophyll. Upper surface of the leaf has palisade mesophyll. Palisade mesophyll is the type of mesophyll by which they are tightly packed. What is in the lower area, just under the palisade mesophyll, you can see another type of mesophyll. You can see it is called spongy mesophyll. As the word indicates, spongy, and you can see large number of air spaces are there. Intercellular space, air spaces are there. And you know, and why the lower surface is having air spaces? You know that upper epidermis and lower epidermis. Lower epidermis, in between there are some minute pores you studied. No, that is nothing but stomata. Stomata is responsible for transpiration, as well as it is essential for gaseous exchange during photosynthesis and respiration. And we are discussing the photosynthesis, right? So that's why you can see the upper surface of the leaf is more darker due to palisade mesophyll, and the lower surface of the leaf is less darker and due to a spongy mesophyll. So palisade mesophyll and spongy mesophyll. So that's why which area the sunlight is falling on the upper surface, right? And that's why the reason the area the sunlight is falling on the upper surface. So that's why upper surface of the leaf, my dear students, it is more green in color. Upper surface of the leaf is more green in color, and it is due to which type of mesophyll, my dear students? It is due to palisade mesophyll. And you know, palisade mesophyll tightly packed, more chloroplast, more green in color. Okay, so they're all connected, right? And what about lower surface? Lower surface, spongy mesophyll, less amount of chloroplast, less green in color, because sunlight is directly falling on upper surface. So that's why the leaf is having an adaptation like that. Okay, and you know the third one, my dear students, you know there are a lot of adaptations adaptations are there, and I'm going to mention only 4, my dear student. What is the third one? You know that when you are looking on the top of from a top of a tree, you cannot see the ground, you can see. For example, you know, when you're flying on the aeroplane, when you are looking, the area you can see full of a canopy, a tree canopy you can see. You cannot see the bottom or the floor of the forest region. The reason is very simple. What is that? The leaves you can see. The leaves are arranged perpendicular to the sunlight, my dear students. Because sunlight is falling. Sunlight is falling means what? If the sunlight is falling vertically, if the leaves are also vertical, what will happen? They are not getting the sunlight right. So when the sunlight is falling in this direction, you can see leaves should be there in perpendicular to each other. Hey, you know, perpendicular, right angle to each other. So that's why right angle 90 degree. You know that. So that's why here you can see the leaves. Leaves are, listen please, leaves are right angle to the solar incidence or solar radiation. So solar radiation is coming, but you can see it is right angles, my dear student. That is an important point, my dear student. Then only what will happen? You can see almost all the sunlight get completely trapped and absorbed. Hey, otherwise what will happen, my dear student? Sunlight is directly falling on the ground. It's nothing absorbed by the leaves, right? Okay, so that is the third one. And what is the last one, my dear students? What is the last one? There are so many adaptations are there. Okay, and here you can see the leaf is having another adaptation. What about the position of stomata? Hey, what about the position of stomata, my dear student? And stomata is also one of the important. If stomata, you just imagine if stomata are located above, what will happen? And you know, due to high temperature during daytime, evaporation, transpiration rate is very high. So that's why that is not good for the plant, right? Because plant, you know, water is inevitable component for photosynthesis. So that's why if stomata are there, if large number of stomata are there on the upper surface, what will happen? More the transpiration rate, more the evaporation rate, that is leading into wilting of the leaves you studied, right? So that is why the reason the stomata are located in the lower epidermis, my dear students, and stomata are mainly located in the lower epidermis to prevent evaporation or to prevent the rate of transpiration, my dear students. And these are the 4 points. At the same time, and there are some, if you need some more extra, my dear students, I can give one more point. For example, what about the arrangement of veins and veinlets on the leaf? The leaf blade called a lamina. You can see the lamina, you can see a midrib is divided into large number. You studied, right? And for example, when you are looking a leaf into account, what is the middle one called what? The middle one is called what? Midrib. And midrib is having the vascular tissue of a plant. Vascular tissue of the plant means what? Vascular tissue you studied of xylem and phloem. Xylem tracheids, xylem vessels, and phloem is having sieve tubes, companion cells. All are there because xylem can conduct water and minerals towards the leaf, the kitchen of the plant, while the phloem is taking all the prepared food and other materials from the leaf to all other parts by a process called translocation. You study, translocation is a process of transferring of food and other materials from the source. Source is mainly leaf to all other parts called source. So source to sink. Source is the area by which the food is stored and the food is produced, and you can see the sink, source to sink, right? Sink means the area where it has to be stored. So this is what we call what? Here you can see translocation. For that, xylem and phloem together called vascular bundle. Xylem and phloem together called the vascular tissue. So vascular tissue is there. And but here this is a broad region called a lamina. Each and every nook and corner of the lamina should get water. Then only they can perform a photosynthesis. Each and every part of the leaf must get all the minerals, my dear students. So that's why the midrib is divided into large number of veins, and each vein is divided into large number of veinlets. So veins, the spreading of veins, the midrib veins and veinlets are providing the nutrients as well as you can see the water and minerals to each and every part of the leaf. So that's why altogether the leaf is well adapted for photosynthesis. That are the points you have to remember in the first question, my dear students. So already I showed one picture here. Here you can see this is the leaf. You can see the broad area. It's already labeled it as leaf lamina. So the leaf lamina is there, the leaf lamina, and you can see the veins and veinlets also you can see. And these are the 4 major adaptations of leaf for performing photosynthesis in a good manner, my dear students. Okay, I hope it is clear, my dear students. Moving without any waste, I'm moving into the next question that is also not an easy one. Why do leaves become yellow in the absence of sunlight? Generally the leaves are green in color, right? And generally the leaves, that the leaves are green in color. And here, my dear students, why the leaves are green in color, you already studied, no? And this is due to a particular pigment is there, my dear students. What is the name of the pigment, my dear students? It is chlorophyll. You keep your mind, dear students, at some one activity related point we studied one thing. What is that? There are some leaves are there, or a potter plant we are taking with variegated leaf. One experiment we studied to prove that chlorophyll is essential for photosynthesis. Variegated leaf means what the area is not having chlorophyll, that's why it is not green in color. But you know that only chlorophyll is present in leaves as their photosynthetic pigments, my dear students. No. Generally, in grade 10 we are studying chlorophyll is the pigment which is responsible for photosynthesis. But along with the chlorophyll, my dear students, along with the green colored chlorophyll, other non-green colored pigments are also there. And I am repeating that sentence one more time. Along with the green colored chlorophyll, there are some non-green colored pigments are also there, my dear students. And I will give one example for that. One is called xanthophylls, my dear students, and another one is called carotenoids, and etc. There are so many, my dear students. For the time we are giving only two. Xanthophylls and carotenoids are the different types of pigments. Xanthophylls and carotenoids are the two different non-green pigments present. So that's what happened. If sunlight is not there, because the question is absence of sunlight, and absence of sunlight means what? If sunlight is not there means what? You take a potter plant and put it in a dark room. Hey, take a potter plant, a green colored leaves, put it in a dark room. And dark room means there is no sunlight, my dear students. After 1 or 2 days, you can see the wilting of the leaves because they cannot perform photosynthesis. At the same time, you can see the green color is gradually changed. Why there is a change in green color? Because you can see in the absence of sunlight, my dear student, the chlorophyll undergoes breakdown. Hey, that is the most important. The chlorophyll undergoes breakdown, my dear students. The chlorophyll undergoes breakdown means what? The chlorophyll in its natural form, it is green in color. And when it is degraded, my dear student, chlorophyll is degraded means what? Chlorophyll is completely gone. And chlorophyll is completely degraded means what? The non-green colored, and especially you can see yellow colored pigments are dominant over. Dominant receive, you study, And here in a green colored living plant, you can see chlorophyll is the dominant one. That's why green in color. And whenever the chlorophyll is degraded, and whenever the chlorophyll is degraded, you can see the non-green colored pigments are dominating. Non-green colored pigments means what? Generally yellow in color, my dear students. So this is one reason for making the green colored leaves into the yellow colored leaves in the absence of sunlight. It's a 1 mark question, so that's why only that point. Degrading the chlorophyll pigments, and that's why the reason the other non-green colored pigments named xanthophylls and carotenoids are dominating, my dear student. That point you can write. Okay, and what is the next question, my dear students? What is the next question? And here you can see, name the digestive enzyme. So after the autotrophic nutrition, now we are moving into the next one. You can see the heterotrophic nutrition. Okay, so heterotrophic nutrition, one simple question is there. What is that? And there is name the digestive enzymes and their source gland. Okay, so some students are reading only the first part of the question. Name the digestive enzyme. They are writing only digestive enzyme. No, my dear student, the question is name the digestive enzymes and their source gland. And source gland for the digestion of the following. And 3 different types of food materials are given. One is called egg yolk. You can see egg yolk. Egg is an edible material. We are all taking egg. You can see, and that egg, the outer shell is not edible. That we are removing. And after that, a white color region is there, and that is what we call what? Egg albumin. In the middle, you can see egg albumin. And egg albumin, as you all know, that egg albumin is mainly made up of. First of all, you have to identify what type of food is given, my dear student. And the food which is given is broadly classified into 3 different categories: carbohydrates, proteins, and fats. And in the topic heterotrophic nutrition, you studied how carbohydrates are digested, how proteins are digested, how fats are digested. So you have to write the points depending upon the mark. But if it is 1 mark question or 2 mark question, 3 mark question, your answers means what? The pattern is different. For example, 1 mark question is not there, only MCQ. The 2 mark question means what? Point wise you can write. While the 3 marks and 5 marks question, that is descriptive type, you have to write in detail. And here you can see the egg yolk, that white color one is called egg white or egg albumin, which is the major content is nothing but protein, you can see. Well, you can see the egg yolk, you can see. Egg yolk is yellow in color, and here you can see it is the major content is nothing but fat, you can see. The lipids and the fats are there in egg yolk. And finally, the milk sugar. As you all know that, what is the milk sugar? The milk is a balanced diet with the different types of nutrients. Milk, carbohydrates are there, proteins are there, vitamins are there, all the nutrients are there, minerals are there in milk, you can see. And here the question is not milk sugar, you can see. The milk sugar is, as you all know, the lactose is the milk sugar. Lactose is the milk sugar means what? Lactose means what? It is carbohydrate. Already here sugar is there. Anyway, you can see this is what we call what? The 3. And here you can see the egg yolk that consists of fat, and egg albumin that consists of protein, and milk sugar that consists of carbohydrate. And here one by one we can discuss about your name the digestive enzymes. And first of all we can see the fat. Fat, you know, one thing is that always you can see the fat, you can see fats are there in large fat globules you studied, and large fat globules are not directly digested. For example, after taking the fat itself, the fat is entering into a stomach. From stomach, it is entering into the small intestine means what? And the small intestine is receiving a duct from liver. And you know, after that, they are contacting with the bile juice. You know that a concentrated bile juice, it is already coming from the gallbladder. You studied, right? And here you can see the large fat globules are turning into a small fat globules. You know, we are very familiar with that process, my dear student. This process is called what? Emulsification. And that process is called what? Emulsification of fat. The first one is not fat digestion. First of all, the large fat globules are broken down into a small fat globules, and that small fat globules, other conversion of large fat globules into a small fat globules are together called what? Emulsification of fat. Only after emulsification, listen, then only the enzyme is coming. After the emulsification only, there is an enzyme. That is a question, my dear student. And you know the enzyme is called pancreatic lipase. What's the name of the enzyme? Pancreatic lipase. And pancreatic lipase is the enzyme which is responsible for digesting, not the fat, digesting the emulsified fat. Emulsified fat, my dear student. So the emulsified fat is digested by an enzyme called a pancreatic lipase. And you know that what is the source gland? And here this is the enzyme, my dear student. What is the source gland? The source gland is pancreas. Pancreatic lipase is produced from pancreas, right? Emulsification is done by bile juice, and bile salts are there, and the bile salts is a part. Bile salts are the part of bile juice. Bile juice is produced by liver. But you know here the question is digestive enzymes. Here they are concentrating on enzyme. So that's why the enzyme for the digestion of egg yolk, it is nothing but pancreatic lipase for digesting emulsified fat. And pancreatic lipase is produced from pancreas, and there is their source gland also. They asked, you already studied, I can give one more point over there. And after that, what will happen to the fat, my dear student? Fat is converting into a product. You know that. What is the product, my dear students? What is the product? Actually, it is converting into a product called fatty acids and glycerol. You know, fats are completely digested to form two compounds called fatty acids and glycerol. And fats, first emulsification, after the digestion to form two simple compounds called what? Fatty acids and glycerol. And the next one, my dear student, proteins. And proteins, you know that proteins are digested by here you can see the proteins are digested by, first of all, you know, no proteins are digested in your buccal cavity because there is no protein digestive enzyme. But when it enters the stomach, you can see an inactive pepsinogen is there. You studied, right? An inactive pepsinogen is produced by gastric glands, you studied, and the inactive pepsinogen is activated by HCl, hydrochloric acid. Already acidity is there inside our stomach, right? So that's why that can enzymes, first of all, you know, it is first one is called pepsin, my dear students, and other one after that, you know, when the food using the pepsin, it is not completely digested and it is converted into peptones, or it is not complete digestion is not taken place in our stomach. So in stomach you can see it is by pepsin, and when it enters the small intestine, the pancreas is producing another enzyme called trypsin. Easy to study. Pepsin and trypsin are the protein digestive enzyme. There are so many other enzymes are also, they're not required for great aminopeptidases. You can see carboxypeptidases, no need. And here you can see the pepsin. What is the source gland, my dear student? And pepsin, the source gland is gastric glands, my dear student. Gastric glands are located on the lining of the stomach. Gastric glands are there. That gastric glands are having different type of cells. It's not important. But parietal cells are there. thyroxine cells are there, my dear students. Anyway, here you can see the gastric glands are there, and the trypsin is produced by pancreas. The source gland is also an important one, my dear students. So that's why pancreatic lipase, and this is the enzyme pepsin, this is the enzyme trypsin, my dear student. Pepsin and trypsin are the protein digestive enzyme. First of all, you have to identify what type of food it is, and after that you have to mention the name of the enzyme as well as their source gland. Okay. And here also I am giving that a point, whenever we took a protein, you can see proteins are completely digested by pepsin and trypsin to form the simplest form of protein. It is called what? Amino acids. The yellow colored one, it is not asked in the question because this is a revision time and the last lap you are. So that is why the reason I am giving some more extra. So proteins are completely digested to form amino acids. Amino acids, important, my dear student. There are 20 amino acids are there in our body to make protein. Different. What? We are taking different types of protein food each and every day, and they are completely converted into 20 different categories of amino acids. That amino acids are very important for our growth, our immunity, all our hormones, all our enzymes, all our pigments. Proteins are very important for our body, my dear student. And the next question, and what is the next one? You can see the milk, sugar, carbohydrates I already told, and here you can see what is the enzyme, my dear students. Here you can see the carbohydrates means what? Carbohydrates starts its digestion from the buccal cavity itself, because here there is one enzyme is there, and that enzyme, the name of the enzyme is salivary amylase. Textbook it is given salivary amylase. Salivary amylase is there present in the saliva, and saliva is produced. What is the source gland, my dear student? What is the source gland? And the source gland, that it is nothing but salivary glands, my dear student. Salivary glands. And salivary glands are located in different regions. 3 pairs of salivary glands are there inside your buccal cavity. And the first 2 pairs of salivary glands are located in the side. You can say they are called parotid. Parotid glands are there located on the 2 sides. 1 pair of parotid glands. And and 1 pair of salivary glands are there under the tongue, sublingual. It's not important, sublingual, submaxillary. One is located on the back side, 1 pair is located on the back, and 1 pair is there on the side called parotid. 1 pair is there on the under the tongue called what? Sublingual, and 1 is located at the junction between maxilla and mandible, upper jaw and lower that area. You can see 1 more pair, that is what we call what? You can see the submaxillary. So parotid glands, sublingual glands, and submaxillary glands are the 3 pairs of salivary glands located in your buccal cavity. And among the 3, the largest one is nothing but the parotid glands from the 2 sides. So when I am taking any type of food, you can see the saliva are produced from the 2 sides, from the bottom, from the back, all together. The food is thoroughly mixing with the saliva to kill the germs as well as for digesting the carbohydrate. But carbohydrate digestion is also not completed in your buccal cavity, because we are not keeping our food for a long time in our buccal cavity. Before chewing itself, we are directly swallowing, my dear student. So you have to keep chewing the food properly. If you chew it properly, the food is thoroughly mixed with the saliva. Almost all the germs are killed at the same time. Almost all the carbohydrates are converted into simple sugars. Complete digestion is not taking place. And what is the remaining, my dear student? And no carbohydrates are digested in your stomach, because stomach is not having any type of carbohydrate digestive enzyme. And here the second one is called what? Pancreatic amylase, my dear student. Easy to study. Just like pepsin and trypsin, you can study salivary amylase and pancreatic amylase. Pancreatic amylase, the source gland is called what? Pancreas. As you all know that pancreatic amylase, the source gland is called pancreas. So here you can see, you keep in your mind one thing, all types of food get completely digested in your small intestine, because then only after digestion, what is the next one? Absorption. Absorption takes place from the small intestine using the finger-like projection villi. So that's why, and here also I want to add, what is that? You studied, my dear student, all the carbohydrates foods are digested to form the simple form of carbohydrate, that is what we call what? Glucose. And here, this is the detailed idea about, 3 different types of food. Only 3 different types of foods are there given in your text. You can see one is called a fatty food, other one is called a protein food, and the last one is called a carbohydrate food. And the first of all, here I am writing emulsification, then only digestion. Here direct digestion, here direct digestion, and here you can see the fats are broken down into fatty acids and glycerol. And egg albumin or proteins are digested to form amino acids, and here milk, sugar, glucose. And these are the digestive enzymes and their source gland. In addition, we study the final products of digestion in that particular context. I hope it is clear, my dear students. Okay. And what is the next one, my dear students? So nutrition is over. Moving into the next one, what is that? Differentiate between tidal volume and residual volume. You are very familiar with what? The word residual volume, my dear, because it is given in the textbook. Tidal volume is not given, but residual volume is given. So you know what is residual volume? You studied already. So residual volume is generally represented by RV, residual volume, and you know the tidal volume is always represented by the letter TV. So TV is the tidal volume, residual volume the RV, my dear students. So already RV is there in the textbook. So that's why you should study the another term. You know the another one which is related with the residual volume, that is called tidal volume. First of all, I will you are familiar with RV. First of all, I will explain about TV. What is TV, my dear students? For example, when a person is under rest, I am not under rest now. Anyway, you can see a person is under rest. For example, you just imagine I am under rest, and for the amount of air I am breathing in and breathing out. Listen please, the amount of air, because you know I am standing in a room with air, right? That air I am breathing in, right? All of you listen please. I am breathing in, and after that, breathe out. Breathe in, breathe out. The amount of air breathe in or the amount of air breathe out. I will repeat one more time. The amount of air breathe in or the amount of air breathe out is called what? The tidal volume. So what is tidal volume? So tidal volume is the amount of air breathe in or out during rest, my dear students, not during a heavy exercise, during rest you can see. So amount of air a person can inhale or exhale under rest, that is what we call what the tidal volume. It is approximately 500 ml. 500 ml means what? Almost half liter. So when I breathe in, half liter of air is getting in. What is that? Is it TV or RV? It is TV, tidal volume. And when I am breathing out, I am eliminating or I am exhaling what we can say again the same amount, that is called 500 ml, that is called a tidal volume. So what is tidal volume? Tidal volume is the amount of air a person can breathe in or breathe out during the normal respiration. During the normal respiration means what? Not a forceful one. Okay, and here you can see what is RV, that you studied. No, what is that? It is also the amount here, tidal volume, residual volume common word is there. So that's why it is also the amount of air remains in our lungs, my dear students. Hey, it is not. RV we cannot take in, we cannot move out. RV is always there inside our lungs, you can see. RV, the residual volume, is the amount of air. The amount of air remains in the lungs even after a forceful exhalation. Even after a forceful exhalation. For example, generally I am exhaling normal exhalation. A forceful exhalation. A forceful exhalation means what? I am exerting pressure to eliminate that air. That is called forceful. Even after forceful exhalation, the amount of air remaining in my lung, that is what we call what? RV, my dear students. This RV is essential for exchange of gases, my dear students. Always some amount of air is always required in our lungs, my dear student. Otherwise, what will happen? So when I breathe out, complete air is gone. And when I breathe in, complete air is getting in, means what? That is our lungs are not getting proper time for exchange of gases. So that's why there should be enough time for exchange of gases, my dear. And you know how can you provide that enough time? By providing RV, the residual volume. Okay, so residual volume and tidal volume, chance for asking, especially residual volume is a short question, tidal volume is an additional one. Okay, so TV and RV, that are the two different types of volume. Okay, what is the next one, my dear students? What is that? And this is a tricky question. What are the muscles involved in the mechanism of breathing? So when I am breathing in and when I am breathing out, what are the muscles present in my body? So most of the students are writing one answer. One is called what? Ribs, and other one is called a diaphragm, my dear students. And these are the two topics we already studied. What is that? Ribs are. So when I am breathing in, what will happen? Ribs are lifting upwards and the arch-shaped diaphragm moves downwards. You studied, right? What is that? There are two types of movement take place. What is that? Look at my hand, my dear student. For example, my right hand is diaphragm. What will happen to the diaphragm? The arch-shaped diaphragm become flat. Hey, what about the ribs? Ribs are moving forward. What happened inside? Inside the pulmonary volume, the volume increases. Automatically, what will happen? When the volume increases, the pressure decreases. So that's why pressure is low. What about outside? Outside pressure is more. Hey, so outside pressure is more, inside pressure is less. You already studied air is always moving from higher pressure area to lower pressure area. So that's why air is coming from outside to inside. That is called breathing in. Hey, this is not the question, the mechanism of breathing. So mechanism of breathing, I mentioned about breathing in. What about breathing out? Ribs are moving backward and ribs are moving backward and the flat diaphragm become arch-shaped and moves up. So what will happen? The volume decreased. And volume decreased means what? Automatically pressure increased. Volume and pressure inversely proportional. So volume decrease means what? Pressure increased. More pressure here, less pressure outside. Air is moving from high pressure area to low pressure area. The breathing out. So you studied, the ribs and diaphragm are the two. But keep in mind, diaphragm, my dear student, already made up of muscles, right? So that's why whenever you are writing diaphragm itself, you are given the mark because diaphragm is made up of muscles. Hey, diaphragm is a muscular organ, so that's why it can be an arch shaped, it can be moved down, it can become flat, and as well as it can move up. Everything is done by that particular diaphragm itself is a muscular organ. So that's why when you write diaphragm itself, you are given half mark. But what about the ribs? Ribs are bones, right? How the ribs can move forward and up, backward, or how the ribs are moving, lifting upward and downward, my dear students. That is not possible directly with the ribs, my dear student. Because, for example, if these are ribs, my dear student, this is one rib, this is another rib, my dear students. All of you look at the board, please. Here you can see, in between the ribs, you can see there are some muscles. Hey, that is important, my dear student. Here the question is, what are the muscles involved? Not what are the organs involved. What are the organs involved means what? If you write ribs and diaphragm, you will get the mark. But here you can see diaphragm is a muscular organ, so that's why no need to write separate muscle, and the diaphragm itself is the answer. But ribs, if you write ribs, ribs are not a muscular organ, it's a bony structure you study, a rib cage you study, right? So here you can see, my dear students, all of you, here you can see in between the ribs, look at the board please, in between the ribs, there is a special type of muscles, my dear students, and in the red color what I am showing, a special type of muscles. A special type of muscles are arranged between the ribs, my dear students. Between the ribs. So that's why I am writing the name, my dear students. Intercostal muscles. What is the name of the muscle? Intercostal muscle. So in between the ribs, you can see intercostal muscle, my dear students. I will write one more time. Intercostal muscles, my dear student. And intercostal muscles are the muscles which are located between. And here you can see there is external intercostal is there, as well as internal intercostal muscles are there. That's not important. At least you have to study that the movement of the ribs are facilitated by, repeat that sentence one more time, and the movement of the ribs are facilitated by the muscles between the ribs. The muscles between the ribs are called what? Intercostal muscles are there. Intercostal muscles are of two types: external, the red color, outside, and internal, inside. And external and internal intercostal muscles are responsible for the movement of ribs, and the diaphragm itself is a muscular organ, and these are the two different types of muscle. What is that? The diaphragm is made up of muscles, as well as the ribs, in between the ribs, internal and external intercostal muscle. So that is the answer for that particular question, my dear students. Here, to get the clear cut idea, I'm showing a diagram, my dear. So the white color region, that is nothing but the ribs, you can see, and this is the rib, my dear student. This is also the rib. In between the two rib, you can see a color region. What is that one? That is the intercostal muscle. Here, the white color region, the ribs, you can see. So our lungs region, it is not only the rib cage only, no. In between the ribs, you can see intercostal muscles are there. And that intercostal muscles are of two types: external intercostal and internal intercostal. So that will give you the clear-cut idea about how the ribs are lifting upwards as well as lifting downwards, my dear students. Okay, so that's about the topic. And the next one, my dear students, I think this is one of the frequently asking question, my dear student. And design a flow chart. So whenever you are asked to make a flow chart, some students are just writing word by word. No, but reflex arc in control and coordination, you studied a flow chart. Like that here also a flow chart. How can you make a flow chart? Flow chart means what? Appropriate boxes are there. So that's why before moving into the flow chart, first of all, you should know the diagram of double circulation. Here you can see this is the diagram of double circulation. You can see one side of your heart. So actually already I marked over here. This is the left side of the circulatory system, and this is the right side of the circulatory system. In the middle you can see a four-chambered heart, you can see. And four-chambered heart, because the question is human being, double circulation is found in human being. Double circulation is there in mammals and birds. You studied a four-chambered heart, the warm-blooded animal. And what about the amphibians and reptiles? Amphibians and reptiles are cold-blooded animals, and they are having incomplete double circulations because they are having mixing of blood. They are having three-chambered heart. What about the fishes? There is no mixing of the blood, but fishes, you can see two-chambered heart. They are cold-blooded organism. And here, we are discussing about the double circulation. Here you can see the left side, why red in color, why right side fully occupied blue in color. And it is for representing the different types of blood, my dear students. And the first one, this left side is purely oxygenated blood, and the right side purely deoxygenated blood. And we can start from the lungs itself. These are lungs, right? These are lungs, right? So when we have to start the double circulation from the lungs, and we should end our process of double circulation in the lungs itself, starting from lungs and ending in lungs. And what are the parts? you can see the lungs are having a type of capillaries are there. What are the capillaries, my dear student? What are the capillaries? Pulmonary capillaries. So we can make a flow chart, my dear students. All of you, please listen. We can make the flow chart. Okay, listen please. The first one we have to start with the lungs. So start with the lungs means what? Flow chart. When you are making flow chart, my dear students, you should do one thing. You should make a box. Okay, and after the lungs, I am putting an arrow. And when I'm putting an arrow means what? This is the lungs, right? From the lungs, you can see oxygen is there. That oxygen is taken by the blood capillaries which are located close to the lungs, you can see. And can you give me the name, my dear students? That is what we call what? Pulmonary capillaries. Important, my dear students, pulmonary capillaries. So that's why from the lungs you can see that oxygen is taken by pulmonary capillaries. And here you can see in the pulmonary, for example, if there's a lungs, the pulmonary capillaries are shown just like large number of branching and one cell thickness you studied, right? And that lungs it is taken. And after that, what will be the blood? Actually, blood is oxygenated, right? Hey, and actually the blood, pulmonary capillaries are there in the area. They are taking oxygen means what? The blood is oxygenated blood. And the oxygenated blood means what? Oxygenated blood, you can see. The oxygenated blood is moving through a channel called what? That should enter into the heart, right? And this is the oxygenated blood. Oxygenated blood should enter into the heart, right? Can you give me that? And heart, you can see the left atrium is the chamber. And what is that area, my dear students? That channel is called what? Pulmonary veins. Not veins, more than one. Pulmonary veins are there, my dear student. So repeat one more time. From the lungs, you can see the oxygen is taken by the pulmonary capillaries to make oxygenated blood. Oxygenated blood is moving into the pulmonary veins. Pulmonary veins. Veins are the blood vessels moving towards the heart. Veins are the blood vessels that are moving towards the heart, and whether it carry oxygenated or deoxygenated, that doesn't matter. But you can see the blood vessels that are moving away from heart is called what? Away from heart arteries towards the heart, it is called what? Veins. It is moving towards the heart, right? And it is moving into which chamber? Here you can see from the left ventricle, my dear students, it is moving to which chamber? It is moving into the chamber of the heart called what? Left atrium. Left atrium is also called what? Left atrium is also called what? Left auricle, you can see. This is the left atrium. From the left atrium, it is moving into the left ventricle, my dear students. And here the left atrium to left ventricle, in between you can see a valve is there called a bicuspid valve or mitral valve you studied. And here you can see, so left atrium, left one. And here it is the responsibility of this one to distribute the blood to all the body part. So that's why the largest artery is located in that region, my dear students. What is the largest artery, my dear student? The largest artery is called what? Aorta, systemic aorta, but at least 10th standard aorta is enough, my dear student. And aorta is dividing into arteries, my dear student. The biggest artery is called what? Aorta. Aorta divides to form arteries. So here you can see this is the major systemic aorta or aorta. Aorta is dividing into arteries, my dear student. Aorta the largest one, and so many branches are there, and finally they are making arteries, my dear student. And arteries, you can see they are, whenever entering into your body part, whenever entering into the tissue, you can see again it is dividing into capillaries, my dear student. What type of capillary, my dear student? And they are developing into another type of capillary called what? Tissue capillaries. This is the method of presenting a double circulation. I started from the lungs, but here I change the color because the blood is moving through the heart. That's why I change the color, my dear student. And blood is moving through the heart, and the first time the blood is moving through the heart in the form of oxygenated blood, you can see that's called left atrium and left ventricle. And after that, the largest artery called aorta. From aorta to the arteries. Arteries are again divided to form tissue capillaries. Already one capillary we discussed, pulmonary capillary. And here you can see this is the pulmonary capillary. So repeat one more time. Lungs, pulmonary capillaries, pulmonary vein, left atrium, left ventricle, aorta, arteries, and tissue capillaries. After tissue capillaries, what will happen? Exchange of materials take place. What is that? Oxygen is given into the tissues for cellular respiration. You study glycolysis as well as Krebs cycle aerobic respiration. You study after that the waste material of the cellular respiration called carbon dioxide is taken. So oxygen and carbon dioxide are exchanged between capillaries, my dear student. And here after that the tissue capillaries, and the blood is changing into oxygenated, is changing into deoxygenated. The amount of oxygen is reduced, right? So here and from the tissue capillaries, it is moving into a structure called vein. So arteries are coming into the tissues, divide to form tissue capillaries, and after the exchange of material, and they are developing to form another structure called vein with the deoxygenated blood, my dear students. And the veins combine to form the larger veins are there called what? Vena cava. And here you can see this is what we call what the tissue capillaries. Tissue capillaries are changing into veins, and veins combine to form vena cava, my dear student. And that vena cava, there are two vena cava you studied. One from the upper part of the body called superior vena cava, one from the lower part of the body called what? Inferior vena cava. So that's why you can see that two vena cava enter into the right atrium, my dear student. So here I can change the color because the blood is again coming into the heart, right? From the vena cava, it is moving into the part called right atrium. Here you studied left atrium. Left atrium is receiving oxygenated blood from pulmonary veins, while right atrium is receiving deoxygenated blood from vena cava. And from right atrium, it is moving into right ventricle, my dear students. And you know, this is the first time the blood is moving in the form of oxygenated blood, and the second time, because the circulation is not completed, this is right atrium to right ventricle, my dear students. And here you can see this is the right atrium to right ventricle, and right ventricle, a blood vessel is moving away from heart. And what is that one called what? And from the right atrium, my dear students, you can see here you can see right atrium, it is moving into this is important one, my dear students, pulmonary artery. Don't confuse with the pulmonary vein, because it is a blood vessel that is moving away from heart. Pulmonary artery that carries deoxygenated blood. And that pulmonary artery, look at the diagram, you know, this is the pulmonary artery. Pulmonary artery is moving into the pulmonary capillaries again. And here you can see the pulmonary, the last point, my dear students, pulmonary capillary to pulmonary artery to pulmonary capillaries. And you know, when you're drawing the box like that is the right method, my dear students. And what is the last one? From pulmonary capillaries to lungs. So, my dear students, you know, we completed a circulation. It is not a circulation. You started from lungs, and you know, we are ending with the lungs itself. But you know, how many times the blood passes through the heart? And twice. Once here and once here. So, twice means what? Double circulation. In a circulation, 2 times the blood is reaching or passing through the heart. One in the form of oxygenated blood, other one in the form of deoxygenated blood, my dear student. And this is what we call a double circulation. And this is the flow chart. This is the flow chart showing the double circulation. Sure, short question. There may be chance for asking some sub questions from this topic, my dear student. I will repeat one more time. Lungs, all of you listen please, last time please. And you know, the lungs, you can see pulmonary capillaries, pulmonary vein, left atrium, left ventricle, aorta, artery, tissue capillaries, vein, vena cava, right atrium, right ventricle, pulmonary artery, pulmonary capillaries, and lungs, my dear student. And the left side is fully occupied with oxygenated blood with more amount of oxygen than carbon dioxide. And the right side of the heart, that is a deoxygenated blood, means what? More amount of carbon dioxide than oxygen. You keep in mind, oxygenated blood has both carbon dioxide and oxygen, but oxygen is more. That's why it is called oxygenated. And deoxygenated blood contains both carbon dioxide and oxygen. Which one is more? Carbon dioxide is more. That's why the reason it is called deoxygenated. Hey, some students are thinking only one thing. What is it? Oxygenated blood means only oxygen, no carbon dioxide. And deoxygenated blood means what? Only carbon dioxide, no oxygen. That is wrong. You know, it's a mixture of both. Both oxygen and carbon dioxide are there in oxygenated and deoxygenated depending upon the amount, you know, it is called oxygenated and deoxygenated respectively. And I hope it is clear. Double circulation, my dear students. You know, I'm moving into the next one. There is an assertion and reason question. There is chance for asking assertion. Students are always confused with the assertion reason type of question. Among which one? There is no confusion in C, there is no confusion in D. Confusion is always there for A and B. Why? A and B, right? Because you know, if the assertion is wrong, no issue. You can write that it is option D. If the assertion is correct, reason is wrong, no issue. You can write the option is C. But what is the first two, my dear students? What is the first two? What is that? Assertion and reason are correct. Oh my God, that's a problem, my dear student. Assertion and reason are correct means what? It is the responsibility of the student to recognize whether the reason is the correct explanation. If it is the correct explanation, it is B. If it is not the correct explanation, it is called what? If it is the correct explanation, it is called A. If it is not the correct explanation, it is called B. So that's why the students are confusing with that one. So okay, let me discuss one question, my dear student, related with assertion and reason. What is the assertion, my dear student? What is it? Blood capillaries, we discussed. That's why you can connect with what? Blood capillaries have walls that are 1 cell thick. Blood capillaries you already studied. There are 2 different types of blood capillaries you studied. One is called pulmonary capillaries near the lung surface. Other one is tissue capillaries near the tissue surface. surface. These are the two different types of capillaries. These are the two different types of blood capillaries. You know that only the exchange of material take place only in capillaries. For example, look at the board, my dear students. For example, if this is the artery, my dear students, you already studied arteries are thick walled. You studied, arteries are thick walled because blood pressure is very high. You know that blood pressure is very high. So these are arteries with a thick wall. And whenever it reaches near a tissue, you just imagine the group of cells are called tissues, right? When it enter here, my dear students, what will happen? It is divided into large of this type of simple arrangement. You can see compared to artery, they're thin walled, right? Because it's only one celled thickness. One celled thickness means what? If you take one area and magnify it, it will be like this. This is one celled thickness. One celled thickness means what? Diffusion is easy. Exchange of materials are easy. Otherwise, you just imagine what will be the problem. It's very difficult to exchange the gases. It's very difficult to exchange the materials. Not only the gases, the materials are also glucose and nutrients and all are what, including water, all are. So here this is called capillaries, the blood capillaries, and after that what will happen? Exchange take place, my dear student. After that you can see there is another channel is called veins. This is artery, this is vein. What is the difference? You can see arteries are thick walled while veins are thin walled. So that's why here valves are also present in veins because the blood pressure is very less. There is chance for backflow. So this is arteries divide to form capillaries, and capillaries combine to form veins. These are the three different type of blood vessels in our body, my dear students. What are the three different types of blood vessels? Artery, capillaries, and veins. And here the question: blood capillaries have walls that are one cell thickness. The assertion is a correct statement. And what is the reason for that? What is the reason? Exchange of material between the blood and surrounding cell. This is the blood, right? And this is the blood, right, my dear students? So blood is flowing through this area. Blood is flowing through this area, and the blood is passing through this capillaries means what? And the blood contains all the nutrients, everything. And here you can see that material. You can see here. You can see when the material is passing through this one, what will happen? Exchange of material. So exchange of material between the blood and the surrounding cell. This is the blood, the red color, and the surrounding cell white in color, and the blood and surrounding cells take place across the blood capillaries. And there is no exchange between artery and tissues. There is no exchange between veins and tissues. Exchange occurs only between capillaries and tissue. Since it is one cell thick, the exchange of material is very, very easy, my dear students. So that's why we can say that this statement is also correct. What is the reason? Because blood capillaries are one cell thick. Why one cell thick? What is the reason for that? Because exchange of materials between the blood and the surrounding cells are taking place only through that particular surface. So that's why it is one cell thickness. So when I am repeating this sentence itself, you will be getting the answer. Is it A or B? And as you all know that the assertion and reason are correct, as well as you can see assertion is the correct explanation for a reason. So that's why A is the right answer, my dear students. I hope it is clear, my dear students. And moving into the next question. Hey, that is an interesting question, my dear students. Why do breads... And every day, or what you can say, we are taking breads, right? And bread, that you already studied bread is related with what? Bread is related with a carbohydrate food, right? And carbohydrate, wheat, flour or something, bread we are making. And the bread, it's a product from carbohydrate. So that's a bread. As well as, cheese, you can see. And cheese is prepared, cheese is prepared mainly by bacteria. There are so many fungus or fungi are also there for making cheese. But basically the Swiss cheese, you are very familiar with one cheese. And you can see one picture, my dear students. And this is a Swiss cheese, my dear students. And the question is, what is the question? Why do breads, when you are getting a slice of bread, when you are getting a slice of bread, when you are looking that bread, you can see there are some gaps, you can see. What are those gaps? The holes are there in the bread. And the Swiss cheese is very clear. This is the Swiss cheese and prepared by some bacteria. Propionic bacteria is studying modern. And here you can see that, and you can see large number of holes are there, my dear students. What is this holes actually? To understand that one, you should know that, and which living organism can prepare all these things. You studied, my dear student. It is nothing but the organism is called yeast, that. And yeast is the living organism. Yeast is the fungus which can make what we can say the bread. You can see baker's yeast. We are commonly using the term baker's yeast. And here you can see what is undergoing. You studied the respiration process of yeast. You studied. What is that? What is that? Is it aerobic or anaerobic, my dear students? It is anaerobic. What is the anaerobic anaerobic respiration in yeast called what? Anaerobic respiration in yeast is called what? Fermentation, my dear students. And you know, anaerobic respiration is called what? Fermentation. Here I am going to write. So whenever there is a question, why do breads and cheese have holes? You know that the reason is nothing but what is the answer? So first of all, we can discuss the chemical reaction. Then after that, you will be get the idea. For example, the bread is having the material called glucose. It is a 6-carbon compound. You know that glucose is a 6-carbon compound. You study, you know what is the chemical formula? C6H12O6. C6H12O6 is glucose, and the glucose, all of you listen please, glucose in the absence of oxygen, it undergo a process called glycolysis, my dear students, and the process take place in cytoplasm to make a compound called pyruvic acid, or otherwise called pyruvate plus. Very small amount of energy is also produced, my dear students, and the pyruvate is a 3-carbon compound, so that's why 2 into means what? One glucose is broken down into 2 molecules of pyruvate, because glucose is a 6-carbon compound. 6-carbon compound is broken down into 3-carbon compound, means what? There are 2 pyruvate, right? And what will happen to the pyruvate, my dear students? And the pyruvate, listen please, the pyruvate, listen please, pyruvate. What will happen to the pyruvate, my dear students? The pyruvate, it is not undergoing aerobic here, because yeast is an anaerobic organism. So that's what will happen. The pyruvate here you can see in the absence of oxygen again. The first step is in the absence of oxygen. Even the second step is also in the absence of oxygen, and you know it take place in an organism called yeast, the baker's yeast, and that process is called fermentation. And that fermentation, the first step is called glycolysis, and the second step is called fermentation. What will be the product, my dear students? There is a product called a C2H5OH, my dear students. It is called ethanol. From the chemical equation, it is clear. C2H5OH means what? It is a 2-carbon compound, and plus you can see there is a gas is produced called carbon dioxide, plus very least amount of energy is also produced. So this is called the anaerobic respiration. You studied the 3 pathway of glucose, and here you can see in the bread you can see this is the alcoholic fermentation take place. Alcohol, ethanol, C2H5OH is there, along with there is a gas produced. What is that gas called? Ethanol is there in the bread, but what will happen to the carbon dioxide, my dear student? Carbon dioxide, since it is a gas, it cannot stay there. Automatically when the gas is having a tendency to move up, right? So when the carbon dioxide is there, ethanol is there in this area, but carbon dioxide is there in the area, and that holes, why holes are made? Due to the presence of carbon dioxide. So when there is such a question, why do breads and cheese have holes, my dear student? And due to the presence of carbon dioxide, you are supposed to write the presence of carbon dioxide, as well as, if possible, you can write the equation of alcoholic fermentation. Then only you will be getting the full mark, my dear students. Okay, and that's about the cheese, Swiss cheese, my dear students. Here an MCQ question, my dear students, an MCQ question, and that is also an important one. Hepato-pancreatic duct. Sometimes you are not familiar with the term hepato-pancreatic, and I will explain. All of you listen please. When you are looking the esophagus, after esophagus you can see the next part is called what? Stomach, you can see. And listen, this is esophagus. Esophagus is also called a food pipe, and for the time we can write esophagus only. And this is the large bag called what? Stomach, you can see. And after stomach you can see the small intestine is starting, right? So small intestine, that this is the part of our alimentary canal, and the stomach is almost located in the left side, because this is the middle part of the esophagus. And here you can see this is what we call what? The small intestine. The first part of the small intestine is called a duodenum. Duodenum is the part of small intestine. For example, I can write over here a small duodenum. The word is not there, so that's why the reason duodenum is a part of the first part of small intestine is called a duodenum. And here, my dear students, just roughly I am explaining. Here you can see, here this is the area. This is the area where, and this is the area where, my dear student, this is the area where your liver is located with the 2 lobes. And liver, just near the liver you can see gallbladder is there, and liver is producing bile juice, and bile juice is temporarily stored and concentrated in the gallbladder, and after that it is moving through a bile duct, because bile juice, the concentrated bile juice is coming through the bile duct. At the same time, here another gland is there, my dear students. What is the gland here? What is the gland here, my dear students? Here you can see another gland is there called pancreas. A yellow elongated gland is there. Pancreas is carrying pancreatic juice, right? So pancreas is Listen one more time. Pancreas is carrying pancreatic juice which is moving through the pancreatic duct, and liver is producing bile juice. It is coming through the bile duct, and the bile duct carries bile juice and pancreatic duct carries pancreatic juice. Both are coming through a common area and common duct, my dear student, and it opens into the small intestine. Hey, and listen please, in the first part of the small intestine, named duodenum, is receiving a duct which is a combination of two. What are the two combinations? One is called hepato. Hepato, the word is related with the liver. You know, liver-related infection, that is called hepatitis. Hepato, liver-related. So, hepato means what? This is the white color one, what I represented. This is called the bile duct, and that is the word hepato. And the pancreas, the word pancreatic. So, hepato-pancreatic duct means what? In the middle, this is the area. All of you can see that this is the area called what? That is the area called what? The hepato-pancreatic duct, my student. Hepato-pancreatic duct can carry both. Hepato-pancreatic duct can carry both the bile juice from liver as well as the pancreatic juice from pancreas, and that duct opens into the small intestine. Which part of the small intestine? Which part of the small intestine? It is duodenum, my dear students. So that's why, what is the question, my dear student? Hepato-pancreatic duct opens into pancreatic duct opens into the area called a small intestine. Small intestine, simply we can say that it is the duodenum part of a small intestine, my dear students. So the answer is, what is the answer for this question, my dear student? The answer is small intestine. It is not into large intestine, not into stomach, not into gallbladder, my dear students. Okay. Next question, my dear students, and you know there is one important question related with excretion. So we discuss some questions from, you know, it is not a complete detailing. Anyway, you can see we discuss some questions related with nutrition. We discuss some questions with respiration. We discuss some question with transportation, and finally, you know, one more topic is that is excretion. And here you can see, name the major nitrogenous waste material produced in humans. Okay, so you know, name the major nitrogenous waste material. So first of all, we should know that our body can produce different types of nitrogenous waste material. Different types of nitrogenous waste materials are produced by human beings, you can see. So not only human beings, when you are considering animals into account, different types of nitrogenous waste are there. So simply we can consider, you know, that the nitrogenous waste material. Listen please, nitrogenous waste material. Basically, almost all the nitrogenous waste are basically produced from ammonia. Ammonia is the chief nitrogenous waste. So you know, the nitrogenous waste I am categorized into three, my dear students, because the first one is called NH3 or ammonia, my dear students, and the first one is called ammonia, and the second one is called urea, and the third one is called uric acid. You are familiar with the terms, my dear students. You know, all contain nitrogen. That's why they are called a nitrogenous waste material. Carbon dioxide is a waste in our body, but it is not a nitrogenous waste material, because carbon dioxide is not having nitrogen, right? So nitrogenous waste materials are the materials which are having nitrogen, ammonia, urea and uric acid. And among the three, which one is the most toxic one? Which one is the least toxic one? The most toxic one is ammonia, and the least toxic one is always uric acid. Okay, the most toxic one, ammonia, the least. So more the toxicity, more water is required for elimination. Least the toxicity, less amount of water is required for elimination. So that's why the reason ammonia, since it is highly toxic, it requires huge amount of water for its elimination. So that's why it is mainly found in freshwater fishes, my dear students. Or simply in 10th standard you can study in fishes, my dear students. Fishes are living in water, enough amount of water is there for the elimination of waste material. So that's why freshwater fishes are having the ammonia as their chief or major nitrogenous waste. What is the second category? Urea, my dear students. Urea is moderately toxic, my dear students, and you know, it is, you know, the major organism. We have to study this one, and human beings, and for our body, our body is not having the most toxic material, not having the least toxic material, but you know, our body is having most toxic material, ammonia. We are converting ammonia into urea, and we are eliminating urea. Our body also, ammonia is produced, but ammonia is not eliminated, because for the elimination of ammonia we need a huge amount of water. So that's why ammonia we are converting it into urea. Then that urea is eliminated. So that's why urea is the major nitrogenous waste material excreted by human being. And ammonia is the major nitrogenous waste material excreted by the freshwater organism. While you can see uric acid, my dear students, there are some organisms, especially you are very familiar with the birds and insects, my dear students. And birds and insects, they cannot drink a huge amount of water. When the bird drink a huge amount of water, they cannot fly, right? So birds and insects, they are not drinking huge amount of water. So that's why their body is having least amount of water for the elimination of material. That's why their body is producing the least toxic material called uric acid. So these are the three nitrogenous waste material present in animals. Okay, and the question is, what is that? Name the major nitrogenous waste material produced in human. And the major nitrogenous waste material produced in human is nothing but the answer is urea. So that's why here I can write the answer is urea is the major nitrogenous waste material produced. Okay, and there is one more question, my dear students, one more question. I think this is clear, my dear students. And one more question is there, my dear students, please. And one more question. Where does it produce, or where is it produced, my dear students? Where is it produced? You know that we are all thinking that urea, excretion. So that's why we are all thinking about kidney, my dear students. It is not kidney. That is always keep in your mind. And the urea, all of you listen please, urea is produced in our liver. So you keep in your mind, kidneys are our excretory organ. Kidneys are our excretory organ means what? It can eliminate. Excretion means what? Elimination, right? And kidneys can eliminate it, but liver can produce the urea. That's an important question, my dear student, because you can say the major nitrogenous waste material is urea, and that urea is produced in liver. And here there is one more question. Where it is produced means the answer is liver. But there is one more question. How? How means what? You have to write the mechanism. In liver, what is happening? All of you listen please. Here you can see the NH3. What is NH3? The most toxic nitrogenous waste material called ammonia is coming into the liver. And that ammonia combines with the carbon dioxide to make urea. That equation is very important. All of you listen please. This equation is not there. And that's why you can see ammonia, the most toxic material ammonia, and they combines with another waste material called carbon dioxide to make a product called, listen please, NH2 CO NH2. NH2CO NH2 is called what? Urea. But you know, as you know that already you studied in chemistry, it is not a balanced equation because here two nitrogen are there, here there is only one nitrogen. So that's why I can put a 2 over here. So 2 ammonia plus carbon dioxide. 2 ammonia plus carbon dioxide, it gives NH2CO NH2. Now also it is not balanced, you can see. And here you can see, here oxygen, there are 2 oxygen here, but there is only 1 oxygen here. So that's why NH2 CO NH2 plus H2O. So that's why ammonia, 2 molecules of ammonia combines with 1 carbon dioxide to form urea plus water. What is the chemical formula of urea? NH2 CO NH2. NH2, nitrogen is there. Okay, so this is a nitrogenous waste material called urea, and that urea is produced from ammonia. Ammonia and carbon dioxide combines to form urea and water. This process take place in the liver. This is called urea cycle. It's not important. Anyway, that process take place in the liver. So whenever a person is drinking alcohol, and automatically what will happen? A person is drinking alcohol means what? Automatically what happen? Liver is damaged, right? You studied on liver cirrhosis. So that's why when the liver is damaged, automatically what will happen, my dear students? And the liver is not, liver cannot convert ammonia into urea. So ammonia is huge quantity deposited in the body. So that's why kidney failure is a big problem, as well as liver cirrhosis is also a big issue, my dear students. Each and every organ in our body are maintaining life, right? That's why the reason we are all living organisms, my dear students. When any one of the process, nutrition, respiration, transportation, excretion, when any one of the process is in trouble means what? And the life is not maintained. Already the chapter name itself a life process. And here you can see, so what where does it produce? It is produced in the liver. How it is produced? This is the chemical equation. And the chemical equation of urea is NH2CO NH2, my dear students. This is the equation, my dear students. Okay, I hope it is clear, my dear students. And that's about the questions related with the life process chapter. So you keep in your mind all the time, my dear students. A living organism always require 4 processes to be maintained properly. 4 processes to be maintained properly means what? Nutrition, respiration, transportation, and excretion, my dear students. Okay, so that's all about the class, and you know that is enough for the day, my dear students. Thank you.